Okay, so we've talked about the structure of membranes and we've talked about diffusion and osmosis. Now I think that we are prepared to talk about the first of the main functions of the membrane, transport. Right? We said that there were two main types of membrane proteins. There's a few variants on it. The first is transport proteins, and the second was receptors. So let's talk about transport and transport proteins, how things get across the membrane. So there are a few different mechanisms by which things can get across the membrane. We've talked about how water gets across the membrane, and we said that there was a special protein called an aquaporin, which is basically a little hole, a little door, that only water can travel through. This is a type of transport called carrier-mediated transport. So that means whenever you have a special protein, that protein is the carrier, and it is what mediates, what allows whatever it is that's being allowed across to get across. And we've already talked about aquaporin, but water isn't the only thing that needs to get across membranes. It's absolutely essential the water gets across, but there are, if water could only get across, there are tons of things that the cell just simply could not do. Like it couldn't take up food, for instance. There are three types of carrier proteins based on, how they're letting things in. Uniporters, uni for one, right? So this is like an aquaporin. It carries one type of solute, right? So there's one thing. It allows that thing to cross the membrane, right? Aquaporins allow water to cross. That's it, just water. Doesn't have anything to do at the same time. So another example of a uniporter is a calcium pump. What does it do? It pumps calcium. It moves calcium from one side to the other. Symporters. A symporter is a carrier that moves two different things simultaneously across the membrane. And it has to be two. It is two or none at all. So let's say that you have something that allows sodium and glucose to get across the membrane, right? It doesn't allow just sodium. You have a bunch of sodium, sodium can't get across. You have a bunch of glucose, glucose can't get across. It's only if you have sodium and glucose, they both cross, and they both cross in the same direction. This mechanism is called cotransport. Many things are taken up by cotransport. A good example of this is the sodium-glucose transport. This is one of the mechanisms that your cells use to move glucose across the membrane. When you have glucose in your blood, your cells need glucose to survive. That's where they get their energy. They actually have a regulated mechanism where they take up glucose and sodium at the same time. And there are reasons for this that we will get into in a second, but basically it allows them to move glucose against its concentration gradient. We'll get into more about how that works. Our third type, if we've got something that carries two or more solutes simultaneously in the same direction, we can also have two or more solutes carried in opposite directions. This is called an antiporter. Sim means same, anti means opposite. So in this case, we have two or more solutes. Again, you have to have them both. You can't have just one. But this time, you have to have one on one side and the other on the other side, and they move in opposite directions. So these are the three kinds of carrier proteins. There are also 3 mechanisms of carrier-mediated transport. Any of these 3 mechanisms can be used with any of these 3 types of carriers, with one caveat that I'll get into. The 3 types of mechanisms are facilitated diffusion, primary active transport, and secondary active transport. I'm going to get into more detail on each of these as we go on. So, first thing that I want to talk about is passive transport. Passive transport is sometimes also called facilitated. Here we are. Facilitated diffusion. Right? So facilitated diffusion, that means it's a type of diffusion. Remember, diffusion is the movement of things from high concentration to low concentration. Things want to diffuse. It is a natural tendency, a basic law of the universe, that things move from areas of high concentration to low concentration. You don't need to put any energy in to allow diffusion to take place. All you have to do is give it a path. You have a whole bunch of things that are highly concentrated in an area. If that area has just like all walls and all the doors are closed, those things can't diffuse, those people can't diffuse out of that room. Open up the doors and the people can leave. All you've done is open up the doors. People want to leave, they don't like being crowded together. So this type of transport requires a special carrier mediator, a protein, a pore, or a carrier protein, but it does not require energy. So let's see some passive transport in action here. So note these. Where is the concentration of red dots higher? On the upper side or the lower side? So there's more of them on the upper side. So which direction do you think they are going to diffuse? Let's see. All right? So now we have an equal number on each side. Now it is in equilibrium, which means that diffusion will take place equally in both directions. The movement across the membrane doesn't stop, but the concentration won't change. Right? So this mechanism is a pore. A pore just makes basically a hole, a doorway that things can move across in. You can also have carrier proteins that instead like bind and shepherd something across, rather than just making a hole. That's often used for larger molecules. That's what this is. Which way do you think the yellow diamonds are going to move across? Remember, it's the type of diffusion. Well, let's see. So here we have, boom, it moves in there, gets shepherded across, and now we have a equal concentration on each side. Now, you'll note that these proteins that I've showed you here, these are uniporters, right? This is a red uniporter. All it does is move the red dots. This is a yellow uniporter. All it does is move the yellow diamonds. Let's take a look at Symporters, right? So this is a Symporter for cotransport. And what you're going to see here is that in order to move across, you have to have something that binds here. See that red slot, or that round slot there? What does that look like it's going to bind? And you're going to have something that binds here as well. It could be these two on this side or these two on that side, but they have to be going the same direction. Take a look at the concentrations of our red dots and our yellow diamonds here. which direction do you think things are going to diffuse? Well, let's take a look. And we see cotransport has taken place. Now, let's go. This is an antiporter. Antiporters take things in opposite directions at the same time. That's countertransport. Again, we have a round binding slot up here. We have a pointy binding spot down there. Take a look at the concentrations, predict which direction things are going to diffuse, and then let's take a look. This is countertransport in action. Boom. And there we go. We see that countertransport has occurred. We've moved two things simultaneously in opposite directions. So far, all of this has been passive transport, right? So you've seen the passive transport with a uniporter, passive transport with a symporter, passive transport with an antiporter. It is also possible to do what's called active transport. Now, passive transport is just facilitated diffusion. It allows things to move whatever way they want to diffuse down their concentration gradient. It does not require any energy. Active transport is the opposite. It moves things against diffusion, moves things against their concentration gradient, and things do not want, naturally, to move against their concentration gradient. It's like pushing a rock uphill. Pushing a rock downhill is easy. Rock takes care of most of the work. Gravity does the rest. When you're pushing a rock uphill, what do you need to put in? You need to put in energy. So here, we're going to need to use energy, and the energy form that is usually used is ATP. And ATP is going to be used directly by the protein. So let's take a look in action here, right? So there's a few things to note here. Note the concentration gradients. And remember that this is active transport, so we're going to be moving things against their concentration gradients. See that? Boom. Goes down, goes down, goes down. Boom. Things can't go back up. You see that little lightning bolt that popped up here? That lightning bolt represented the use of energy. But now we have moved everything to one side. Note that that first one actually went with the concentration gradient. The important thing about active transport is that it uses ATP directly. It uses energy. You can actively transport things down their concentration gradient, and sometimes your body does that if you want them to move faster than diffusion would normally allow them to. But the important thing about active transport is that it uses energy. And it can, and usually does because your body doesn't like to waste energy, it can move things against the concentration gradient. Again, what was this? Think about it. Was this a uniporter, a symporter, or an antiporter? This was a uniporter. It only moved one type of thing. That was primary active transport. The difference between primary active transport and secondary active transport, they're both active, so they both move against the concentration gradient, or they both use energy. But primary active transport uses energy in the form of ATP directly. Secondary active transport is going to use ATP in the form of a stored concentration gradient. So we're going to have actually a two-point process. We're going to move a whole bunch of one thing to one side, and we're going to spend ATP directly to do that. We're going to do primary active transport to do that. Those things are now going to want to diffuse back where they came from. they have a natural tendency to diffuse back where they came from, and we are going to utilize a symporter to force them to carry something else along when they move back across. So let's take a look here. So we've built up a pretty hefty concentration gradient here. You see there's 4 red dots down here. These 4 red dots really want to diffuse back across, but they can't. They can only move the one direction, the way that the active transport pump, the primary active transport, let them. But they do have another way. They can utilize this symporter to get back across. But we know that the symporter can only move things in pairs. So if the diffusion gradient is high enough, we've stored enough energy in this concentration gradient that they really want to move across, they will have enough energy to take one of these yellow triangles against their concentration gradient. So let's see that happen. So this is the equivalent of, instead of like having a like an engine, a motor that's going to directly burn fuel to lift a rock up a hill, right? Instead, what you're going to do is attach that rock to a bucket that's already at the top of the hill. And then you're going to use energy to fill the bucket with water. And when the bucket gets heavy enough, the bucket will go down and it will pull the rock up. You're not using any energy directly on the rock. You're using the energy on the bucket. You're filling the bucket with water. But the bucket, when it gets heavy enough, will pull whatever it is attached to with it. Why would you do this? Why not just have primary active transport for everything? It's basically an efficiency mechanism. Your body only wants to make a certain number of proteins. The more proteins it has to make, the more energy it has to spend doing it, and the less space it has to put those proteins in there, and the more likely it is something's going to go wrong. So if it had a primary active transport mechanism for every single darn thing that it wanted to move across, it would have to make a lot more proteins. Instead, what it does is it just has a primary active transport mechanism for a few things. One of the main ones is sodium and potassium. There's what's called a sodium-potassium pump, which we will talk about in great detail later on in the course. And so you build up a concentration, moving a whole bunch of sodium to one side, and then you can use the energy that you stored in that sodium to bring a whole bunch of things back across. You could use it to bring in glucose, you could use it to bring in calcium, you could use it to bring in hormones, anything like that. Once you've stored the energy in the sodium, it's very versatile. But you only need one primary active transport mechanism. You move the sodium across, and then you just have a whole bunch of symporters that all involve sodium. There's another way to move things across the membrane. So we said that these carrier proteins, the pores and things like that, well, they're proteins, right? They're pretty big proteins, but they're still proteins. What if you want to move something that's bigger than a protein across the membrane? Like, let's say that you are a white blood cell, and you want to eat that bacteria over there. Well, bacteria are bigger than proteins. And clearly, you can't fit a bacteria through a protein. Right? You can't fit something big through something that's smaller than itself. So you need a way to move really big things. And that is vesicular transport. Vesicular transport is used to move really big things across the membrane, or to move a whole bunch of smaller things across the membrane all at once. And the way it does this is, instead of letting things through the membrane, we are going to take the membrane in as well, making like a membrane bubble. So if we have like a membrane here, and let's say that this membrane wants to eat this bacterial cell, right? Then what happens is the membrane kind of forms itself into a bubble surrounding this bacterial cell. And then that bubble kind of closes off, and then it just sort of blorps free, and you end up with a bubble of membrane surrounding whatever it was that you just ate. So a bacterial cell, in this case. That is actually specifically a type of vesicular transport. There are several types, called endocytosis. Right? So endocytosis is when you use this vesicular process to move things into the cell. You're sucking things into the cell. Endocytosis has three types. There are three types of endocytosis. There is phagocytosis, and that is actually what I just showed you there. Remember, phago means mouth or to eat. Cyto means cell. So this is cell eating. This is where you engulf a large particle by growing sort of pseudopods, or these like tentacle things around it, and then you suck that thing inside. Right? This is done by macrophages and other white blood cells called phagosomes. And it's the way that cells eat things that are really big, like cells or viruses. There is also pinocytosis. Pino means to drink. So this is cell drinking. This is actually pretty much the same thing as cell eating, except as phagocytosis, except there's no bacterial cell there. Instead, you're just sucking in a huge amount of water all at once. And it's a way for cells to basically get a whole bunch of water in really, really fast. Faster than aquaporins would let them in. Third method of endocytosis is what's called receptor-mediated endocytosis. And this is the way that you absorb a whole bunch of small things all at once. So in this case, instead of a bacterial cell, you have a whole bunch of receptors in your membrane, right? And each of these receptors is going to bind to something. This could be a food particle, or it could be a protein, or a hormone, or lipids. This is actually a part of how your cells take in like cholesterol and fats and things like that. And so you see here, these blue proteins have bound to the receptors, and then you get basically the same thing happens. You're going to suck in a bubble, and the protein that causes this bubble to form is called clathrin, and so this bubble is sometimes called a clathrin-coated pit. But either way, it has its receptor proteins in there, and then stuck to those receptor proteins you have whatever it was that was bound. and you've just taken in a whole bunch of those things. Now, if you have endocytosis, endo meaning inside, taking things into the cell, you can also use vesicular transport to force things out of the cell. And that is called exocytosis. Exocytosis is the main way that cells are going to release a whole bunch of proteins out of the membrane. Most proteins are too big to move through channels and pores. So you make them in the endoplasmic reticulum, and then they're going to break off in a vesicle and go to the Golgi where they get sorted. And then all of the ones that are supposed to be gotten out of the cell are going to be all packed up into one vesicle, and that vesicle is then going to move towards the cell. And so, like, let's see here, we have, this is a vesicle, and this vesicle is full of these blue proteins. And this vesicle is next to the cell surface. And what it does is it just gets really, really close, and they sort of slam together. If you've ever seen, like, those little, like, kids' toys, you see them at kids' birthday parties, those little, like, things that you dip them in the soap, and then you blow in them, and they make these bubbles. Well, have you ever seen two of those bubbles collide in the air? What happens? Well, you get two bubbles collide in the air, and they kind of go blorp, and now you've got one big bubble. And membranes actually behave kind of like soap bubbles. And so these two things get really close together, and then you sort of slam them into each other, and they form they sort of blorp together. And when they do that, all of these proteins or whatever spill out. And so this is how cells are going to release proteins. It's also how, for instance, nerve cells release neurotransmitters, because it's a really fast way to release lots of things all at once in a big mass. And it's also the way that, like, let's say a cell has eaten a bacteria, and now it's got a whole bunch of waste products. That is all of the parts of the bacteria that it couldn't digest. Well, it's got to get them out. Exocytosis is the way it's going to get rid of them. This vesicular transport always requires the use of lots of ATP. So it's a form of, technically, active transport. It takes lots of ATP to move these things, but you're moving lots of things. So it's less efficient in terms of energy than carrier-mediated transport is, but it allows you to move things that would be too big or move things faster than could be done with carrier-mediated transport. So here we just basically see a picture of those. This particular picture is receptor-mediated endocytosis. So you can see a whole bunch of receptors on the cell surface. These receptors bind this whatever is out there, this extracellular molecule, whatever they're transporting across. You form a pit, this invagination, in the cell membrane, and you can actually see that forming on an actual picture of an actual cell up here. And these dark proteins here that are kind of pulling that pit in are proteins called clathrins, and so we sometimes call this a clathrin-coated pit. And then it sort of wraps itself around and blorps off, sort of the reverse process of when you have two soap bubbles run into each other. And then you just blorp this whole thing away, and you now have a vesicle that contains whatever it was that these receptors bound. Exocytosis, again, the reverse. You've got your vesicle full of whatever it is that is going to be secreted. These are going to be released from the cell. That's a process of secretion, so these are called secretory vesicles for secreting, and They just run in together. There are these linking proteins that force the membrane and the vesicle close together. The vesicle and the membrane merge, and that allows everything that's inside the vesicle to just sort of zip on out. And here you can see an actual picture of that happening. This is exocytosis. Exocytosis and endocytosis can be combined. This is a process called transcytosis, and it does happen in your body, particularly in, say, your stomach. Well, not really your stomach, your intestines, right? Your stomach doesn't really absorb many nutrients, but your intestines do. So in your intestines, you need to absorb lots of nutrients. Those nutrients need to go to the blood cells. Your blood cells don't run directly into your intestines, because if they did, you'd bleed out your guts all the time. So you have a layer of cells that are going to be in between your interstitial fluid, your internal fluid where your blood vessels run, and the lumen of your intestines where all of your food is. And what happens is you take in a bunch of food particles through receptor-mediated endocytosis, but those don't need to go into these cells lining them. Those cells don't need that food, or at least they don't need most of that food. Most of that food needs to go into your blood. So those vesicles that were just endocytosed over here just cross the cell and then are exocytosed onto the other side. But the only things that can then get through are going to be those things that bind to the receptor and get endocytosed. This process of endocytosis, transporting the vesicle across the cell, and then exocytosis is called transcytosis. All right, so that is the way things move in and out of cells. To rehash this, the take-home message, you need to know the 3 types of carrier proteins: symporters, antiporters, and uniporters, and you need to know what the difference is between each of those. You need to know the differences between passive transport or facilitated diffusion, primary active transport, and secondary active transport. You need to know what vesicular transport is, and the 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. You also need to know the exocytosis and transcytosis and what each of those things are.